Welcome back, everybody, for a new unit. This time around we're going to be talking about mitosis, which is the process of cell division. You see, as a cell gets older, for a variety of reasons, it may need to divide and reproduce itself to go from one cell into two cells. And this is vital because all throughout your life, every single second, there are millions and billions of cells in your body that are dividing and having to replace themselves. When we grow, we need new cells. When cells get broken or damaged, we need new cells. When cells wear out, we need new cells. And the process of mitosis is how we get those cells. Now, mitosis can happen for a variety of reasons, but first we should talk about exactly what it means. Simply defined, mitosis is just cell division. One cell becoming two new cells. Now, the new cells we call daughter cells. They're like the little babies of the original cell, and they are identical to each other. So if you started with a nerve cell, you end with two new nerve daughter cells. If you start with a muscle cell, you end with two new muscle cells. There's not a particular special name for the cell they come from. Sometimes it's called a parent cell or original cell, but it just means a mature adult cell. And there are lots of reasons mitosis can happen. Anything that might cause a cell to need to replace itself or to multiply. So as you get bigger, growth, bigger, stronger, your muscles grow, your bones grow, your nerves grow, so you need more cells to make them up. Mitosis, it's how we get more cells. Another way you might get more cells, or reason you might need them, is to repair themselves. Just like any machine, a biological machine like our bodies can break or run down, and we have to fix the parts that are broken. So when you cut your skin, for instance, your body may need to repair itself by growing new cells to cover up that old damaged tissue. Finally, what can also happen, another cause for mitosis, is reproduction. We need to get new cells to replace the old cells that wear out. Some cells have a natural lifespan to them. Muscle cells, nerve cells, any other tissue will eventually just wear out and break. It goes through too much wear and tear. And when that happens, we have to replace it somehow. And that somehow is mitosis. Now, off to the side here, you can see a diagram or a series of pictures of a cell undergoing mitosis. We've got our original adult cell. You see it sort of bunches itself together. And through a process we're going to explore in this lesson, the one cell divides into two new cells. And right here we have, right before they finish dividing, you can see both of these are just like smaller versions of our original cell. So this is the original. These are the daughter cells. Just like our cells unit, there's a lot of vocabulary in this one. So if you memorize these terms, it's going to help an awful lot towards making sure that you get the grades you want on the test. So let's go through them. DNA, we know, is the part of the cell that carries information. But DNA has a lot of different structures it can have. Chromatin is what we call DNA when it's uncoiled. It's just loose in a spiral inside the cell, hanging out, doing its thing, which is great when the cell needs to get to the DNA. Unfortunately, sometimes the cell needs to just store the DNA and not have it floating around, not have it in the way. And so, a chromosome is what we call a length of DNA when it has been coiled up. So instead of having that really long bit of DNA stretched out like we saw before, instead we coil it really tightly like an old-fashioned telephone cord, or like Christmas lights after Christmas. You coil them up nice and tight so they don't take up that much room, and it prevents them from tangling with anything else. Let's get rid of that so it doesn't get in the way and you can still read everything. Okay, now that chromosome contains a lot of genes, which are just, for now, we'll call them segments of DNA. They're lengths of DNA that do a job. So maybe from here to here might be a gene. It's not really that simple, but for now that's all you need to know. The centromere. So it has this great context word in there, centro. Sounds like center, right? That's because it means the middle, the center. The centromere is a region of DNA, so it's just a part of the DNA, typically found near the middle of a chromosome. So when we coil it all up, like we talked about earlier, that DNA has got a middle portion, and where two copies of DNA, which copies of DNA are called sister chromatids, two sister chromatids are two identical copies of DNA. Where those two meet, we have what's known as a centromere. So right up here, this region is the centromere. Not the spiky thing. We're going to talk about the spiky thing in just a second. Just the area that I've highlighted here. So, spiky things. Spiky thing is called a kinetochore. Kinetochore is just a protein. Remember that the body uses proteins to do most of its jobs. It's a protein that surrounds the centromere and holds it together. Again, almost always it's more complicated than that, but that gives us a nice idea about what's going on. If we find our centromere up here, that's this guy. It works kind of like a paperclip. It's holding these two copies together. So to review what we've seen right here, chromatin is DNA when it's all loosey-goosey. A chromosome is when it's coiled up nice and tight. Same thing, just different structure. And when we coil up a chromosome nice and tight, you can kind of see over here there's little lines in here. It's supposed to represent a coiled chromosome. It pairs up with another chromosome that codes for the same information. For reasons which you'll learn when we talk about DNA later on, your body keeps two copies of all the chromosomes. This is because if one copy is damaged, it can use the other one to fix it, among other reasons. Now, the centromere is the region where those two bits of DNA, those two chromosomes, contact each other. This region in here. And the kinetochore, going back to our copies idea, the kinetochore is the paperclip holding it together. So think of these, if you were to write out your genetic code in a piece of paper, you make two copies of it. Each copy we'd call a chromosome. The part of the paper where we staple it, we call the centromere. And the staple itself, what helps hold it together, that's the kinetochore. Okay, a little bit more, we're almost done. Spindle fibers. Spindle fibers are a type of microtubule. That sure sounds familiar, right? That's from way back in our cell structures unit. The microtubules are parts of the cytoskeleton, and these spindle fibers help to pull apart or separate the chromosomes when we divide. So you can see them right in here. Here's our microtubules, or our spindle fibers. They reach out kind of like chains or grappling hooks, and they'll grab on to the kinetochore from one side, and another one will reach out and grab onto the kinetochore from the other side, and then they both tow in their opposite directions, and it pulls those two copies apart. We're undoing the staple and separating our two copies here, because each daughter cell is going to get one of our copies. We've also got, let me change the right color here, centrioles. Been a long time since we talked about those. Those are organelles. They're found only in animal cells, not in plant cells. And what they do is, you can look right here, they help to organize those great spindle fibers. They keep everything under control. So basically they're guiding the process of division. They're what make the spindle fibers do what they need to do. It talks about completing cytokinesis. That's a process we'll see in just a little bit. And finally, we have the aster. Now, aster is Latin for star, and in fact, this cell structure is shaped like a star. Here it is in here in the picture. Here's one, here's another one. So when the cell begins to divide up, we call the two different ends of it poles, just like the North Pole and the South Pole on the planet, or the positive and negative poles of a polar molecule. We get one aster at each side, or each pole of a cell when it's dividing, and those asters are like the center that everything else pulls to. Okay. Nothing to write for you guys on this particular bit, just to see. Remember, the DNA is held inside the nucleus of the cell, and if we were to zoom in on just one of these little X's right here, we'd see a chromosome. Two sister chromatids. Those are the two sides that are identical copies of each other, right? There's one, there's another one. Held together in the middle. The region where they're held together, there's our centromere. And it's not shown in this picture, but the spiky little paperclip that holds it together, right there, that'd be our kinetochore, holding it together. And all of this stuff, all this DNA, remember, all these genes, remember, is DNA. A big, long, huge, humongous molecule. Now, chromosomes, like we were talking about, are DNA. And DNA is made up of genes. A gene is like a special way of saying a length or a portion of DNA that does a specific job. So there are thousands of genes, each of them controlling things like your hair color. Some of them control things like your eye color. They control how tall you are, all kinds of different things about your body. So basically, to help us remember, DNA, that's the main molecule. DNA, when we have a lot of it that is doing a particular job, a length of DNA is called a gene. And when we have a lot of genes all together coiled up to get more organized, we call that a chromosome. Okay, and I kind of drew over the picture because I needed some room, but here's the actual pictures of chromosomes. Used with an electron microscope, we can see them. They really are shaped like little X's. Now the DNA, remember, is a macromolecule. We talked about those before. And the DNA is made up of what we call nucleotides. Those are different pieces that make it. Now of these little chromosomes up here, humans have 46 of them. Different organisms each have different numbers of them. Now 46 is in 23 pairs. As you can see right here, if you count all these guys, we get 23 pairs of sister chromatids. Not important much for this unit, though. What is important is that chromosomes, remember chromosomes, the coiled up DNA, these little X's we're seeing in the picture here, are only visible during cell division. Because other times that DNA stays all loosey-goosey. It's only during cell division that we take this long molecule and coil it up nice and tight. Now before a cell can divide, what it's got to do is make sure that the two new cells will work. And so it's got to copy its genetic material, and yes, I know this is a typo, thank you for pointing it out. It's got to copy its genetic material, so the DNA, to make sure both cells have a full set of instructions, and its cellular material, that's the organelles. We've got to make sure that each of the new cells has mitochondria and ribosomes and all the other things that make a cell work. Okay, chromosomes. This is our last slide for this little bit right here. Chromosome structure, the way a chromosome, or coiled DNA, is put together, is before cell division. So before we even try to divide, we make copies. Here's a copy, and on the other side, here's a copy. You always want to have more than one copy of any important document, and your body feels the same way. And remember, we call those two copies sister chromatids, and they're attached at the centromere. And that's that for this slide and this particular set of notes. Thanks for watching, everybody. If you had trouble with the vocabulary, remember to look at your flashcards and go back and watch this again. After we repeat it a few times, you'd be surprised how quick you pick it up. Oh, sorry.